Good afternoon, children. This is my third video on our lesson Human Reproduction. In the previous video, I had discussed the structure of female reproductive system, various parts, their functions, then various layers in the uterus, various parts of uterus and the other structures like fallopian tube, etc. And in detail, I discussed about the structure of an ovum. Today, we are going to start with our new topic, that is gametogenesis. The term gametogenesis is made up of two parts, gamete and genesis. Gamete means a sex cell, whereas genesis means the formation or the birth, right? So by definition, this process means the formation of haploid gametes, that means N, haploid gametes from diploid germ cells in gonads, that is ovaries and the testes, for sexual reproduction. Alright, the process, that is gametogenesis, is of two types. The first type, that is spermatogenesis, means formation of sperms in testes of the males, and the other type, which means oogenesis, means formation of ova in ovary of females. Today's topic that I am going to begin is only the first type of gametogenesis, that is spermatogenesis. But before we start with the detailed study, let me tell you the basic pattern of this development in both spermatogenesis and the oogenesis is broadly the same, right? It's very similar except for some minor differences. Like we can say both of them, they involve three phases. The first phase we are going to start here would be multiplication, growth and maturation, right? And here also accordingly the phases are multiplication, growth and maturation. So today's topic to begin, children, is spermatogenesis. Spermatogenesis means formation of haploid spermatozoa from diploid spermatogonia, that is sperm mother cells. If you study this equation just now here, what is formed from which substance, this can be framed as a definition. For example, the formation of haploid spermatozoa or the sperms from the diploid spermatogonia or the sperm mother cells, this process is called as spermatogenesis. It usually starts in the body of a male at around 13 to 14 years and continues throughout the life. But in old age, this process declines. Coming further, spermatogenesis occurs in seminiferous tubules of the testes and includes three major events. So if I just remind you this diagram, what we had discussed in our first video, the seminiferous tubules, these highly coiled tubules that are separated from each other by various compartments, right? This is the birthplace of sperms and the process of spermatogenesis occurs over here. So this process, as I said, includes three major events, and the first event is, we can say, it is formation of spermatids. Like look here, if you just talk about the summary like this or from your textbook first, the first event is we are going to form through this flowchart here the study of formation of spermatids, right, from spermatogonia. So this is the first event till here. The second event is formation of spermatozoa, and the third is release of mature spermatozoa, which we call as spermiation. All right. So there are three terms to understand, children. We have those similar sounding of the terms, but they are yet very different from each other. The first process is spermatogenesis, right? The second is spermiogenesis, and the third is spermiation. So when I say spermatogenesis, right, it occurs in the seminiferous tubules, and it will have three stages to study, that is from here till here. This entire process is spermatogenesis, right? The first part includes this first stage up till here, formation of spermatids. Then the second part, what we call as spermiogenesis, will be the change from spermatids into spermatozoa, and then later when they're released, we call them as spermiation. So let us begin every detail of this from the study. So the same thing I have drawn over here. In a brief layout, I can show you the spermatogonia or the sperm mother cells. They are diploid with 46 chromosomes. Then they divide by mitosis, leading to formation of primary spermatocytes. The chromosome number remains the same. The next is first meiotic division. See, just now it was mitosis. Now it is the first meiotic division to form secondary spermatocytes, and now here the chromosome number becomes reduced to half. Then from secondary spermatocytes now occurs second meiotic division and spermatids are formed, right? So the next stage would be differentiation, that is spermatids differentiate by a process called spermiogenesis into spermatozoa or sperms, and then finally they are released by the process of spermiation. So now let us start in detail. To start first from the formation of spermatids. This is completed in 3 phases: multiplication, growth and maturation. In the formation of spermatids, this process, I'll begin one by one, but you have to imagine that this is going on in the primordial germ cells of the germinal epithelium that lines seminiferous tubule. Now again I have to show you this diagram. Seminiferous tubule here, when we studied in the structure of male reproductive system, so this is the seminiferous tubule lining that the germination of these cells we are going to talk about. So this outermost layer of cells that are lining the germinal epithelium, this lining of seminiferous tubule, some cells we call them PGCs, that is primordial germ cells. That is, these are the ones that line germinal epithelium of the seminiferous tubule, right? In this diagram, these are the outermost. Inside the structure, the outermost layer, that is the PGCs or the primordial germ cells. Now, first step, what happens is that they undergo mitosis, and by doing so, they will form undifferentiated germ cells, right? The same undifferentiated germ cells, now these will undergo mitosis again and form spermatogonia or the sperm mother cells, which are diploid. That means 46 chromosomes will be like this, 44 plus XY number of chromosomes. Now then, sperm mother cells or the spermatogonia, right, they divide once again by mitosis, right, to produce a number of spermatogonia. And now these spermatogonia that are formed are of 2 types, type A spermatogonia and type B spermatogonia. See, once again let us revise. First I told you primordial germ cells or the PGCs that are lining the seminiferous tubule, they undergo mitosis to form undifferentiated germ cells, which again undergo mitosis to form the sperm mother cells or spermatogonia, which are diploid, and these again undergo mitosis to form a number of spermatogonia. Then these spermatogonia are of 2 types, type A spermatogonia and type B spermatogonia. Type A spermatogonia, they serve as stem cells. That means they divide to form additional spermatogonia. But type B spermatogonia are the precursors or the ancestors of the sperms or the male gamete. They move towards the lumen of the seminiferous tubules and enter a growth phase. That means just now I told you that if this is the lining of the seminiferous tubule, these divisions are taking place in the outermost cells. So by the time this division has reached this place, now these cells will move into the lumen of the seminiferous tubule, right, and enter a growth phase where they'll now be called as spermatocytes, right? So no more spermatogonia, now you're calling them spermatocytes because they have entered the center of this seminiferous tubule. So what happens with the spermatocytes? Now you can see the first multiplication phase is over. The moment they reach the stage of spermatocytes, they enter another stage, what we call as the growth phase. During the growth phase, a number of things happen. Like number one, the spermatocytes grow in size. Second, they accumulate nutrients. Third, they replicate the DNA and are now called primary spermatocytes. Primary means the first formed spermatocytes. And here, a major difference is that these are double the size of spermatogonia. You can see them. So spermatogonia, whatever their volume, here they become just the double of these spermatogonia, right? Then now the growth phase is also over. By the time they have entered this stage now, primary stage, they will be double the size and are called as primary spermatocytes. Now they enter the third phase, what we call as the maturation phase. Each spermatocyte, that is, you're talking of one single, the fate. You can imagine there are millions of them. So each spermatocyte undergoes first maturation division, which is a reduction division. And by this, two haploid daughter cells are formed. One with 22 plus X and the other with 22 plus Y chromosome. And now these are called as secondary spermatocytes. Alright, so they undergo the second maturation division and 4 spermatids would be formed. So what can we broadly say, children? That from each primary spermatocyte, 4 haploid spermatids are formed. That means starting from here, from the primary stage, if one primary spermatocyte is dividing, then the net production is finally of 4 spermatids as the end product. Alright, so this is the first phase, what we call as formation of spermatids. You can see spermatids have been formed by 3 phases. Once again, the multiplication phase, where major division was mitosis, and we ended up forming the spermatogonia. 2 types I told you, A and B. They will be functioning as the stem cells. That is, they will divide and form additional spermatogonia. Whereas the spermatogonia of type B, they are the precursors of sperms, and then they divide in the serial arrangement like this. Now, the stage when type B spermatogonia has formed the spermatocyte, this division from here to here is called as spermatocytogenesis. Spermatocytogenesis, that means it has formed the spermatocytes, which further now grows into the growth phase and forms the primary spermatocytes, which are double the size of spermatogonia. Now these primary spermatocytes, they form the secondary spermatocytes, right, which are half the chromosome number. They're haploid and they end up after second maturation division into 4 spermatids. So this was the first part of this division. Coming to the second part now, we will talk about formation of spermatozoa or sperm by a process called as spermiogenesis or spermatoleiosis. In this, what happens? Nothing much, but it is a total transformation. That means a radially symmetrical spermatid. See, this is a sperm. From nowhere, it's looking like an adult sperm. So, the sperm, just now it's looking like this, radially symmetrical spermatid. It will totally transform into a new structure which is polarized, right? So this polarized spermatozoa, when it has been changing or transforming, a number of changes have occurred. Like number 1, the nucleus shrinks. 2, acrosome formed from Golgi body. This is the acrosome, the tip of this, which contains an enzyme, hyaluronidase, right? Then the axial filament is formed from the Golgi body. Axial filament is the filament inside. I have explained this already in the previous video, student. This filament is the axial filament inside the tail. So that forms from the here. This filament, that is the axial filament, is formed from the distal centriole of the spermatid. We studied in the previous that there are 2 centrioles. One is the proximal. This will gain entry into the ovum, whereas the distal here This is going to form the axial filament. Then what is the next change that is going to happen here is the mitochondrial spiral that is to be formed. This mitochondrial spiral that is formed is in the form of a sheath called as Nebenкern, right? And much of cytoplasm is lost, and the remaining sheath is called as manchette. I'll show you here in the stages shown into the book at page number 111. You can see the Nebenкern, that means this mitochondrial sheath which will be becoming spiral gradually, called as Nebenкern, is formed, right? And finally the thin cytoplasm. The cytoplasm becomes very, very less, and now we call it as the manchette. Alright, and this change from the radially symmetrical form to a polarized structure is called as spermiogenesis or spermatoleiosis. So we are not talking of much of nuclear divisions, etc., but here we are talking about the total transformation. But here we talked of many divisions. First these were mitotic and later on meiotic. Alright. At this stage, now the sperms, they have already taken up their shape, and the head of spermatid gets embedded into Sertoli cells to obtain nourishment like this. These heads of sperms, they have penetrated and entered these Sertoli cells, you can see, and the total purpose is nothing but obtaining nourishment. This is the Sertoli cell, you can see here. So this nourishment that takes up from the Sertoli cell. Coming to the last stage of release, the mature spermatozoa or sperm cells are now released from the Sertoli cells. Where? Into the lumen. That is, from here now the sperms will be released into the lumen of the seminiferous tubule before their passage to the epididymis. And this release is called as spermiation. Alright, mature spermatozoa or the sperms are released from the Sertoli cells. Why were they attached to Sertoli cells? To obtain nourishment. But now they are ready for release. And where are they going to be released? First, just now, into the lumen of seminiferous tubules. And this process is called as spermiation. After spermiation, where are these to be carried? They will be carried to the epididymis to be stored for some time to gain maturity and also mobility. Right? So this is all about the changes that take place in the gametogenesis under the male system, that is spermatogenesis. So try reading this, children. In the textbook it's given in a similar manner, but I'm explaining it more like an equation form, flowchart wise. So you should be able to understand, but try reading it twice and thrice, then only you'll understand it more clearly. Then the last thing for today is the hormonal control of spermatogenesis. See, spermatogenesis started with the onset of puberty. Just now we said as soon as the boy reaches a stage of 13 to 14 years, the spermatogenesis begins. This is initiated by certain hormones, by secretion of some hormones from the hypothalamus. Hypothalamus releases first hormone we are calling GnRH. That means gonadotropin releasing hormone. Alright, this stimulates the anterior lobe of our pituitary, that is the master gland. The master gland is affected by this hormone, and it is stimulated to produce two hormones. The first we call LH or the luteinizing hormone, and the second is called as FSH or follicle stimulating hormone. LH acts on Leydig cells. Remember, L alphabet acts on L only. So the LH hormone affects the Leydig cells, and this stimulates them to secrete the androgens. Androgens means the group of hormones from the male system, and the most important one is testosterone. This testosterone stimulates the process of spermatogenesis because this is the acting male hormone. Alright, so the hormone is released. Then if we talk about the other hormone, that is the Sertoli FSH, these act on the Sertoli cells, the ones that give nutrition to the developing sperms, and it stimulates them to secrete two hormones. First is called as ABP. ABP means androgen binding proteins. And the second is another hormone we call as inhibin. Alright. So ABP, as the name tells, is androgen binding proteins. This causes the concentration of testosterone in the seminiferous tubules, right, for spermatogenesis, right? Spermiogenesis, that is this change. Spermiogenesis, that was the transformation change, is brought about by the APB hormone. Whereas inhibit means to stop. So this name inhibin, it suppresses, that means decreases, right, the synthesis of FSH. Once this is formed, this will inhibit the formation or the synthesis of FSH, which is like a negative feedback. How will it do? Because you can say like inhibin suppresses FSH by stimulating the anterior pituitary here. It will be like a negative feedback, right, affecting the anterior pituitary. Thus we can say that the normal release of testosterone is under the negative feedback. So once again children, it's important that we revise this before we end. First I told you hypothalamus released the hormone called GnRH, that means the gonadotropin releasing hormone, right? This stimulated the anterior pituitary to produce two gonadotropic hormones, LH and FSH. LH is acting on the Leydig cells and stimulates the secretion and synthesis of androgens, that is the testosterone, right? This androgen testosterone is very important to stimulate the process of spermiogenesis, and the entire spermatogenesis is effective under the male hormone, that is testosterone. Whereas FSH, this acts on the Sertoli cells and stimulates them to secrete two hormones further, ABP or the androgen binding proteins. They cause the concentration of testosterone in seminiferous tubules for spermiogenesis, that is a transformation from radially symmetrical form into polarized structure. Whereas the second hormone, that is the inhibin, right, this suppresses the synthesis of FSH, right, and this by the anterior pituitary, how it affects it and suppresses the synthesis. And overall we can say the normal release of testosterone is under the negative feedback control. So that's all for just now children. You have to read the text for today from your pages 109, this page number 109, 10 and page number 111. I have only explained these 3 pages, but because they are all about the psychological parts and many terms we have discussed, they are all very, very important children, and I'll sum up what important terms you should be able to define. First is gametogenesis. Here is the definition, right? I told you how you can explain this in your statement, just this change. The second term is what is spermatocytogenesis. Then what is a primary spermatocyte, how it is different from secondary spermatocyte, right? What is a spermatogonia, here you should be able to explain. Then what is spermiogenesis or spermatoleiosis, and finally what is spermiation. So all this you should be able to explain in definitions and this hormonal control, like which is going to affect what part of the male reproductive system and in what way. That's all for just now children. Any queries, please ask anytime on the group, but before that please read the text twice and thrice and then ask as many questions that you can have. Thank you.